In order to test the experiment, in order to test the hypothesis, we need to do experiment. And after we use a if-then logic to the deductive reasoning to design the experiment, then we also need to have something called control. Control versus experimental group. This is a very important concept. Experimental group is something that we scientists don't know the result. That's why we need to do experiment to find out the result, to test it. We call it experimental group. Control is something that we know the result. Scientists know what will happen if we do the experiment. Then we expect, we are very certain and expect the same result will come out. So control is something that we scientists are very certain to happen. Versus experimental group. Experimental group is the one that we don't know the result and we want to test them. And there are two types of control, positive and negative. Later in this semester in the lab, you will test different types of food using a different type of test. One type of test is called Benedict test. Benedict test tests for glucose, or we call it reducing sugar. So if the food that you test contains sugar, then Benedict test will give you, let's say, Benedict test, the Benedict solution originally is blue color. If you have glucose in it, then it will turn red, or it will turn to different color. If you have a little bit sugar, it will turn to green. If you have more sugar, it will turn to yellow. If you have even more sugar, it will turn to orange. If you have a lot of glucose, then it will turn to red. So it depends on how much glucose you have. So in this case, if you put glucose directly into the Benedict test solution, it will for sure give you positive result. That's why we call this positive control. That's why we call this a positive control, because glucose will give you positive result. On the other hand, if you put water into Benedict solution, well, because water has no glucose in it, we know for sure that the Benedict solution will not turn to different color. It will remain in blue color. So we call this a negative result. And that's why we call this negative control. So water is a negative control, and glucose is a positive control in Benedict test. So this is only Benedict test, and in the experiment later in this semester, you not only will test glucose, you also test starch, you would also test lipid, fat, and so on. We have different types of tests that you can use, but Benedict test is only used for testing glucose or reducing sugar. So let me give you an example. I'll give you three test tubes. So one test tube, it will be glucose solution. Another test tube will be water solution. And then the third test tube will be, let's say, an onion. Onion smashed into water and become an onion mash solution. So we don't know if the onion mash solution will contain sugar or not. So the onion smash solution will be our experimental group. The glucose solution in the test tube will be the positive control. The water will be the negative control. So in this case, the experimental design will be perfect or complete, because you have the onion smashed. solution as your experimental group that you don't know whether or not it has a glucose or not, and then you have the glucose solution as your positive control, and then you have the water solution as your negative control. So this is how we design experiment. Yes, we first use the inductive control to set it up, and then we have to figure out an appropriate positive and negative control. The purpose, so why do we need positive and negative control? Because the unknown, the onion smash solution, the onion mesh solution, if it has glucose, then we can compare the result with the positive control and the negative control tube. If the result of the onion test tube looks similar to the positive control test tube, then we can say that, okay, the onion contains glucose. On the other hand, if the onion test tube the result of the onion test tube looks like the water, the negative control, then we can conclude that the onion does not contain sugar. In other words, the positive control and negative control is for us to compare the result from the experimental group. And in our lab, you will have more than one experimental group, but we will have positive control and negative control for you to compare. So what is a conclusion? Conclusion is an analysis of the data to determine if the hypothesis is supported or not. Analysis of data. Data here is basically the result that we get from doing experiment. Data is not necessarily number, although most of the data are numbers. Most of the data are numbers, but it may not necessarily be number. For example, the previous example I told you in the last slide, the Benedict test. The data of the Benedict test will be the color changes of the Benedict solution. So the data that you need to take, you need to record in your notebook, in your lab notebook, is that is it red, orange, yellow, green, or blue? So this will be your data. The conclusion from one experiment may be used for hypothesis. Yes. After you find out the conclusion of the experiment, if in a real life situation, real life research situation, usually after we finish one experiment, it'll lead us to ask question to do a follow-up experiment. This is what it means in here. But if the results do not support the hypothesis, then we need to form a different hypothesis. So it goes to the flow chart that I told you, that I showed you before. So this is what it talks about. If the result matches the hypothesis, then we need to repeat the experiment until we get consistent result. For example, in LA, if we do let's say the Benedict test to test for glucose, then we get let's say onion. We find out that onion contains sugar in LA. What do? Then we may go to SF again to do this experiment and see if the onion from SF contains sugar. And then we keep repeating, and then we go to let's say Tokyo University and ask the people there to repeat this experiment to see if it get it correct. Then we go to Oxford University and repeat the same experiment. We keep doing this until the whole world, all scientists in the world, they got the same result. The onion contains sugar. Then we can conclude that in here to form the theory, which is, well, onion contains sugar. On the other hand, let's say if we do this experiment and the result or the data is not consistent with the hypothesis, what happened? Then we have to keep on revising the hypothesis and redo the experiment and so on and so forth until when? Until the result matches the hypothesis. Then we can go through this loop here, the repetition in here. So in real life experiment, most of the time we are in this stage. We keep on revising experiment because the result does not match our hypothesis. But once we find out that, okay, the hypothesis matches the result, the data matches the result, then we will be very happy. Usually at this time, we will immediately publish it. Because once you publish it, you will be the first one who discover that, and then the whole world will repeat your experiment. The whole world will repeat your experiment because based on the paper that you publish. And if they can get the same result, then you will become a theory. Then you become a prominent professor. You will be famous, and university will hire you. But if other people, they repeat your experiment and they get this point, the result does not match the hypothesis, then they will suspect, okay, what did you do in your experiment, you know? And then you need more people to repeat your experiment to see if they can get the same result. And if they cannot get the same result, then you cannot form theory. When you cannot form theory, then when you cannot form theory, then you do not become rich and famous, then you cannot be a professor. So that's what happened in academia. What is scientific theory? Let's talk about this here. Scientific theory is something that is well supported. As I mentioned previously, if the entire world repeat your experiment and they all got the same result as you do, then your finding will become a theory. It is supported by lots of observation, experiment, and data. Basically, you may think about theory as the fact, the truth, or even the law. So when you in the lab, you learn about cell theory. What is cell theory? Also in physiology, we also talk about hemostasis. And most importantly, most of you have heard about evolutionary theory. The reason why scientists call it evolutionary theory, because evolution has been well supported by lots of data and observation. So in the scientific community, we all agree that, well, I'll say most of the scientists agree that evolution is a fact, is a law. So evolution is a confirmed theory, it's confirmed. I understand that many of you are religious, and believe it or not, I also have my religion. I would not say that I'm religious, okay? When you take this class, it is required by the school that we teach you evolution. And you are also required to learn evolution in order to pass the exam. So if you are a religious person, I suggest you to bear with it for this semester. After you learn evolution, you get A from this class, and then guess what? You can forget about this and continue to be your faithful servant of your religion. So yes, we will cover evolution, but I ask you to try not to argue with me or try not to argue with each other about evolution if it is against your religion. So we learn this as a student. I teach you as a professor. So that's my position here. Even though I am, well, evolution is also against my religion, but I still teach evolution to you because it is required by the school. So as a student, if evolution is against your religion, you also need to learn about this, and I hope that you get an A as well. So the theory of evolution is a new unified concept of the biology. This is what I just mentioned. Is a well established. It's a law. So it is true. We prove that it is true.